Welcome back, biology lovers. Today we are diving deep into the fascinating world of cellular metabolism as we decrypt the mysteries of glycolysis. So buckle up and join me as we uncover the secrets of glycolysis together. The term glycolysis comes from a Greek word glykis, which means sweet or sugar, and lysis means breakdown. So glycolysis means breakdown of sugar. Here for sugar it means glucose. So if we define correctly, glycolysis is a series of enzymatic reactions which breaks one molecule of 6-carbon glucose into two molecules of 3-carbon pyruvate, C6 to 2C3. It is also called EMP pathway because the pathway was elaborated by these scientists Gustav Emden, Otto Meyerhof and J. Pernes. Now the question is, why glycolysis is a pathway, not cycle, like we know Krebs cycle or you say nitrogen cycle. So the difference between a pathway and a cycle is that in a pathway, the initial compound is converted to a final product by sequential reactions, but in case of a cycle, substrates and the initial compound are continuously regenerated. Imagine the wheels of a cycle always going round and round. Now, the site of occurrence of glycolysis. Glycolysis occurs in the cytoplasm of cell. It is a unique pathway because it does not involve molecular oxygen. This pathway can be found in both aerobic and anaerobic organisms. That's why glycolysis can be called as the universal pathway for glucose catabolism. Phases of glycolysis. Glycolysis is a 10-step process including 10 enzymes, but it is divided into two phases, 5 reactions each. These phases are named as 1. Preparatory or investment phase and 2. Payoff phase. In this video, I am covering the first phase of glycolysis. Preparatory phase. The first 5 reactions of glycolysis is called preparatory or investment phase. So why first phase of the glycolysis is called in such way? Because substrates of this phase consume energy to convert glucose into 2,3-carbon sugar phosphate. As energy is invested, that's why it is called investment phase. Step 1. In the first step of glycolysis, glucose is converted to glucose 6-phosphate by the enzyme hexokinase in the presence of ATP and magnesium ion. So in this picture, you can see glucose has 6 carbons and phosphate group is transferred to the 6th carbon of glucose and hence named glucose 6-phosphate. Here the enzyme is hexokinase. If we recall, kinase is a type of transferase enzyme. Transferase enzymes transfer groups from one compound to another. Here it is transferring phosphate group from ATP to glucose by converting ATP to ADP. The term hexa means 6. As the groups are transferring in 6-carbon glucose, that's why it is named hexokinase. And yes, it is an irreversible step. That's why there is one arrow in this reaction. Glucose 6-phosphate cannot form glucose with the help of the same enzyme. Importance of phosphorylation. So now the question is, why do substrates need to be phosphorylated first? There must be a reason, right? So the importance of phosphorylation in simple terms is, first, cell membrane is impermeable to charged molecules and also it lacks transporters for phosphorylated sugar compounds. So once phosphorylated, glucose and other glycolytic substrates are trapped inside the cells. Second, negative charge of phosphate repels the negative charge of cell membrane. because cell membrane also has phosphates in the form of phospholipid. And the third is phosphates also lower activation energy of enzymes, making them more specific towards substrates. Step 2. In the second step, glucose 6-phosphate is converted to fructose 6-phosphate with the help of enzyme phosphohexoisomerase. Glucose is an aldose sugar containing aldehyde group, but fructose is a ketose sugar containing keto group. So this is an aldose-ketose isomerase conversion reaction. Step 3. In the third step, fructose 6-phosphate is converted to fructose 1,6-bisphosphate by the help of phosphofructokinase 1 enzyme. In this step, 1-phosphate is added to the first carbon of fructose 6-phosphate. It is the first committed step of glycolysis. Before this step, glucose 6-phosphate and fructose 6-phosphate have other fates. But when fructose 1,6-bisphosphate is formed, glycolysis is doomed to happen. 1 ATP is used in this reaction. This is the rate limiting step of glycolysis. Here you can see it is written fructose 1,6-bisphosphate, not fructose 1,6-diphosphate. But we know that bis and di both signifies 2. The difference between bis and di is that bis means 2 molecules attached to 2 different molecules. Here phosphate group attached to 1 and 6 number of carbon. In the case of di, 2 molecules attached to the same molecule like adenosine diphosphate. Step 4. This is the reaction where 6 carbon compound will be clipped to form 2 3-carbon compounds. Fructose 1,6-bisphosphate is clipped to produce 1 molecule of glyceraldehyde 3-phosphate and 1 molecule of dihydroxyacetone phosphate, better known as DHAP. They both are 3-carbon compounds. It is a reversible aldone condensation reaction. Aldose enzyme helps in this reaction in the presence of zinc ion. Step 5. DHAP rapidly converted to glyceraldehyde 3-phosphate with the help of triose phosphate isomerase enzyme. This is a reversible isomerization reaction, just like step 2. So now there will be 2 molecules of 3-carbon glyceraldehyde 3-phosphate. This is the overview of today's learning. By this, our first preparatory phase of glycolysis is over. Payoff phase will be in the next video. If you have any queries regarding today's topic, don't forget to comment below. If you enjoyed it, give a thumbs up, share with your friends, and hit the subscribe button. Stay curious, learn more, and see you in the next video.